Transcription is a process in which RNA is synthesized from DNA molecule with the help of an RNA polymerase enzyme. This is a DNA molecule which would be transcribed into RNA molecule. This DNA molecule have two strands which are anti-parallel to each other. It means one strand is in 5' to 3' direction and the other strand would be in 3' to 5' direction. So one of the strand of DNA which is in 3' to 5' direction will act as a template for the synthesis of single-stranded RNA molecule which is always in 5' to 3' direction. Here you can see the structure of DNA molecule having two strands in which adenine bond with thymine and guanine bond with cytosine. This double-stranded DNA molecule unwinds and in which one of the strand will act as a template strand for the synthesis of RNA. This 3' to 5' direction which consists of T, A, C, G nitrogenous bases acting as a template and synthesize the RNA. And in RNA, uracil is present instead of thymine. So here you can see DNA molecule, DNA strand having thymine bond with adenine, adenine bond with uracil, cytosine with guanine and guanine with cytosine. So this is a strand of RNA molecule. Let's see how transcription happens. This is the first step of transcription which is also known as initiation phase. These two are the strands of DNA molecule and this DNA strand consists of structural region, promoter region and terminator region. Green portion is the promoter region, blue is the one from where transcription will start. So you can see promoter region consists of -35 sequence which is TTGACA box. Why it is known as -35 sequence? Because it is located 35 sequences away from the transcription starting point. -10 is TATAT box because it is located 10 sequences away from transcription starting point. So these -35 sequence and -10 sequences are present in prokaryotes. And these are the promoter regions. These are basically binding sites for the attachment of RNA polymerase. RNA polymerase recognize these -35 and -10 sequences in promoter region, bind here and then start or catalyze the further process of transcription. In eukaryotes, sequences are different. In eukaryotes, -25 and -70 sequences are present which are Tata box and Cart box. So one strand is coding and the other is template. Template strand is the strand from where RNA will synthesize. So this is the starting point of transcription from where transcription will start. RNA always forms in 5' to 3' direction. These two are the strands of DNA molecule which are separate from each other. This is the RNA polymerase enzyme which is attached at the promoter region. This RNA polymerase consists of 4 subunits, 2 alpha, 2 beta subunits and 1 sigma factor. Sigma factor is responsible for the correct initiation of transcription process. And once the transcription has started, it would be removed and the remaining part of the enzyme moves to the template strand and complete the transcription. The remaining part is 2 alpha and 2 beta subunits which are known as core enzymes. So they read the DNA and form the RNA. These core enzymes and sigma factor whole is known as holoenzyme. This is the second step of transcription which is known as elongation phase. Here RNA strand elongates. These two are the strands of DNA molecule. Here the transcription bubble is in the middle where DNA strands are unwinded. So if the DNA strands are separated from each other, this is known as transcription bubble. And this transcription bubble forms when RNA polymerase binds at the promoter region. One strand of the DNA is known as template strand. This is also known as antisense strand. Antisense because it is complementary to the messenger RNA. It is opposite to the messenger RNA. And rewinding of DNA occurs at the back of the DNA. And other strand is the coding strand. This strand is known as sense strand. It is same to the messenger RNA. And unwinding of DNA occurs in forward direction of DNA because RNA polymerase is moving along the DNA in forward direction. So unwinding will also occur in forward direction. This is RNA molecule which is in 5' to 3' direction, and here RNA polymerase is adding the ribonucleotides to the strand of RNA, and these ribonucleotides are complementary to the template strand of DNA. So these are the nucleotides which always add it at the 3' hydroxyl group, so that RNA strand would move in 5' to 3' direction. So these are the ribonucleotides which would be added by the RNA polymerase at 3' hydroxyl group. This is the elongation phase. This is the last step of transcription. This is known as termination. In termination, RNA synthesis stops. RNA synthesis stops by two methods. One is row-dependent termination, other is row-independent termination. In row-dependent termination, row factor is used. Row is a protein which binds to DNA and RNA and releases the RNA and RNA polymerase enzyme from DNA and stops the synthesis of RNA. In row-independent termination, a hairpin loop forms on messenger RNA. You can see like this, a loop-like structure forms. This loop-like structure is because of GC base pairs, series of GC base pairs which are present on mRNA, and they form a loop-like structure which is known as GC hairpin. And at the end of this loop, polyurate is present. You can see on DNA adenines are present. These adenine bind with uracil on RNA, so polyuracil, small polyurate tail is present at the end of the GC hairpin loop, and this is known as hairpin loop. So that's all about transcription. Here transcription will end, RNA synthesis will stops, and RNA is in 5' to 3' direction. I hope you like this video. Please like and subscribe FMZ Biology.